We are live, everyone. So my name is Jesse, and I am with Exploring by the Seat of Your Pants. For those joining us for the first time, we are all about bringing conservation, adventure, and science into classrooms around the world. And of course, there are no classrooms right now. All of you are stuck at home. So we so appreciate you continuing to tune in as we highlight amazing scientists, explorers, and organizations from around the world. Today, we are joined by a new speaker and a friend of mine who I've worked with in a huge variety of contexts over the last 2 years. So we are joined by graduate student Farah Qaiser, and she is a scientist, science communicator, and science advocate who does work she's done every form of science communication you can possibly imagine. But in addition to that, she works on using DNA sequencing to better understand complex neurological disorders, which is so far beyond my understanding it's to be comical. But I'm so excited to hear her talk about it today, share it in a way that's hyper accessible as always, and find out a little bit about the amazing work that she does. Without further ado, thank you so much for joining us today, Farah, and take us away.
Awesome. Thank you so much for having me, Jesse. I am going to open up my slides. And yeah, good morning, everyone. I'm really excited that you're all tuning in from wherever you might be in the world. And today I want to talk to you about DNA, kind of you and your genome. So as Jesse kind of mentioned, so my name's Farah. I am a scientist, I'm a science writer, and I'm a science advocate. Those are kind of a lot of things, so I am busy all the time. But today I'm going to kind of tune in and just zone into what I do most, which is kind of research and looking into DNA. So I want to start off with cells. I'm going to move Jesse over to the corner. So I want to start off with cells. So as you might know, cells are kind of the building blocks of our body. And there's so many different types of cells in our body. So you've got skin cells, you've got cheek cells, you've got nose cells, you've got nerve cells, and just so many millions of cells that make up our body. Now our cells move, they grow, they divide, they die, sad note, but our cells do die, unfortunately. And in order for our cells to do all of these different things, they kind of need instructions to know how to move, grow, build proteins, and so on. And all of these instructions can actually be found in a small little piece of the cell called the nucleus. So the nucleus contains instructions that cells need to grow. Specifically, these instructions are found in something called DNA. So that's deoxyribonucleic acid. Now, there are a couple of things I kind of want to show you about DNA. And the first is that if you look at the shape of DNA, you kind of see this double helix twisted ladder. So it's kind of two strands of DNA which wind together to make this little characteristic shape. So, and as you can see, like each step of the ladder is made up of different letters or bases. So there are 4 bases in your DNA. There is A for adenine, T for thymine, C for cytosine, and G for guanine. These 4 letters make up the steps in your ladder, so they will bond together and they will make each step. So you can kind of imagine like a little person walking up your DNA, and on each step they will see these different bases. Now you're probably thinking, why are you calling these bases letters? Like, just call them bases, Farah. Well, I always like to picture these bases as letters in our DNA, and I'm going to explain why using one of my favorite examples, which is Harry Potter. So I'm going to turn over and read the first letter, well, the first sentence of Harry Potter to give you an example. So, chapter 1, The Boy Who Lived. Mr. and Mrs. Dursley of number 4 Privet Drive were proud to say they were perfectly normal. Thank you very much. First sentence of a great book series. If you haven't read it, you should read it now. But what I really wanted to kind of point out here is that notice that there are different letters. So you've got your M, R, A, and D. And these letters make up words. So you've got Mr. and Mrs. and so on. And these letters make words, which make this entire sentence. So kind of keeping that in your mind, that's kind of how DNA is. So the way letters make up words in a sentence is how bases, so A, T, C, and G, will make up the genes in our DNA. Kind of wild, right? So you can kind of picture your entire DNA as a book. It's like your entire book. And I've kind of got a question for you, so you can feel free to drop it in the chat, which is, how many letters do you think are in our DNA? Do you think it's something like 100, 200, a million, 2 million, 1 Harry Potter book, 2 Harry Potter books, so on? I'm going to give you a minute. Jesse, do you have any ideas on how many letters are in our DNA?
In the whole genome, I think a lot of Harry Potter books, more than the series, more than J.K. Rowling could put out.
That's a good guess. Pretty close, although not close enough. I'm going to hope that people have dropped in answers, and the answer actually is there are over 3 billion letters in our DNA. That's actually 30,000 copies of the entire Harry Potter book series. Like, I've read Harry Potter a million times, but that's not enough to make up for entire DNA. So that kind of just shows you how large our DNA is. That's how many letters and words and sentences there are. And the thing is, scientists are actually using DNA sequencing to kind of go through your entire book, like chapter by chapter, to search the entire book and look for different changes. So if we're still imagining our DNA as a book, you can kind of think that maybe there are missing words, or there are jumbled up sentences, or perhaps torn out pages, missing chapters, and so on. So there are a lot of different changes that can happen. And with 3 billion letters, you know that typos are going to happen. So what scientists are trying to do is they're using DNA sequencing to find these different changes. And they're trying to work out, does every change in our DNA have an effect? This isn't actually true. So some changes can cause nothing to happen. Some changes can have an effect. So scientists are still trying to wrap their head around how different changes can lead to different effects. So for example, my research, I'm trying to use DNA sequencing to better understand complex neurological disorders. Specifically, I work on 2 disorders. So one of them is epilepsy. So epilepsy is a disorder where individuals have multiple seizures, which may last for a few seconds or to many minutes. Then there's autism spectrum disorder, which is the second disorder where individuals may have kind of repetitive behaviors, where they may face difficulties in communication or talking. Now, the interesting thing about these 2 disorders is that people with epilepsy also have autism spectrum disorder, and people with autism spectrum disorder also have epilepsy. So I'm kind of curious, like the parts where they overlap, so the people who have both epilepsy and autism spectrum disorder, can I use DNA sequencing to perhaps understand more about why this happens? So that's kind of me in the lab. Outside of the lab, I kind of enjoy science writing and standing up for science, and I wanted to share a few examples of what that can look like. So this here is the Toronto Science Policy Network. We are a fun group of graduate students. I did co-found the group in July 2018. And what we're basically doing is kind of creating a space for students. So students like you, undergraduates, graduate students, professors, faculty, staff, anyone who's interested can kind of jump in and learn more about and engage in science policy. I know science policy sounds like the vaguest words you've ever heard of. So that's kind of how science can inform decisions. So how science can decide, hey, maybe we should go into quarantine so that we can help fight this pandemic. Or maybe, hey, we should give more money to building this vaccine for the coronavirus. So kind of using science to help with decision making and making decisions to help support science. That's kind of what science policy is. But this group is not just me, but it is full of so many wonderful people who help me do all the events and all the crazy ideas that I sometimes have. So that's one thing that I like to do outside the lab, is kind of take part in this Toronto Science Policy Network. Another thing is that I'm actually over here in Canada, I'm in the Chief Science Advisors Youth Council. So our Chief Science Advisor is Dr. Mona Niemer. She provides scientific advice to the government here in Canada. And she's recruited these lovely 20 people to kind of advise her on different aspects of the science community here in Canada. You can see me in the middle over there, but I am surrounded by incredible people like Justine, who is passionate about plastic pollution, not population. We have Taylor Morriso, who is very passionate about diabetes, Molly Sung, who is a friend of mine, who is a chemist, now a PhD, and is working in Vancouver. We've got Keely Aird, who I think has spoken as Exploring by the Seat of Your Pants before, maybe, yes. And she works with STEM Kids Rocks. She works on bringing science to the youth. So a lot of incredible people here. And the last thing that I do enjoy doing is writing. So I'm gonna pull that off. When I started university, I've always loved reading and writing. So for me, kind of joining my student newspaper was one way I could start writing. So in university, I wrote a lot. I wrote about science. I wrote about how students are doing different things on campus. Like I wrote about anything I could get my hands on and anything the newspaper would let me write about, of course. I think one of my first pieces was actually talking to Jessie, so the person who introduced us on this call today, and how Jessie launched Science Literacy Week, which is kind of this annual celebration of science every year. So that was kind of one of my first pieces, and I'm very excited that Jessie will let me talk to him. So that was also great. I've written about things like selfies. So I know that a lot of you may take selfies right now, but what kind of happens if a scientist takes a selfie? So scientists actually did do a study. We love doing studies on everything, you might have known or realized by now. So in this study, what they actually found was that if scientists take selfies, it actually helps build trust, especially if you can see the person's face in it and the kind of science they're doing. And maybe Instagram selfies could actually be a tool to help build a connection between people. So if you're not already taking selfies, I think you should be, because hey, science has said that it's a good way to build trust. Another thing that I've kind of written about is how women in STEM face barriers. So there are a lot of different groups in science, but not all of them have equal access. So not everyone knows how to get involved in science or how to pursue science. So I kind of wrote about the different barriers and obstacles that women face, and also all the great work that people are doing to help improve this access. And also with all the coronavirus coverage that's been going on, I've also been writing about COVID-19. So for example, in this piece in Forbes, I actually wrote about how the public is going to Wikipedia. So millions of people are turning to Wikipedia to kind of find more information about COVID-19 amid this pandemic. But that's a lot about what I've been doing outside the lab. I wanted to kind of narrow in back to what I was talking about before, which was DNA. So you might be wondering, okay, Farah, you've told me about DNA, you've told me what you do outside the lab. How do kind of those interests connect? So I want to use an example of my science writing to show you how scientists are trying to better understand our genome. So this here is a piece that I wrote on Friday, February 14th, Valentine's Day, but it has nothing to do with love or the heart. It's actually a little macabre, I think. I wrote about Jean-Paul Marat. So Jean-Paul Marat is a French journalist, a French politician, a French scientist. He did a lot of things. Like he has a full biography on Wikipedia. But what's actually quite interesting about Jean-Paul Marat is that in the last few years, he was actually slowly dying, like his health was worsening, but doctors at that time didn't know what it was. They knew he had some sort of skin condition, but they couldn't ever narrow down what exactly was kind of affecting him. But towards the end of his life, what actually happened was Jean-Paul Marat was actually stabbed by a revolutionary. He was actually in the bath at that time because apparently being in the water would help his skin and help him feel comfortable. So we know why Jean-Paul Marat died. He was clearly stabbed to death, as this painting shows, but we don't know what was affecting him in the last few years of his life. Now here's where scientists got smart. So they were like, okay, how can we kind of collect more information? It's kind of always the first step. You know your question. Our question here is, what was Jean-Paul Marat dying of in the last few years? How do we kind of collect that data to help answer that question? So if you look at this photo right here, and if I zoom in, you can see that this little piece of paper actually has blood stains. So it's blood stains from when Jean-Paul Marat was actually dying. So scientists were like, hey, we could grab some of those blood stains. Maybe we could find an answer in there. So that's what they did. They looked at the DNA, they could find those blood stains, but they actually went one step further. Their idea was, okay, so we could look at the blood that Jean-Paul Marat has left on the newspaper, but maybe that blood also contains perhaps DNA from bacteria or viruses or fungi that were kind of affecting his skin. So what if we looked at all that DNA, we matched what we could to Jean-Paul Marat, but we also tried to identify what bacteria or fungus or viruses were there. So that's what the scientists did. They kind of were searching for any bacterial or viral DNA, and they wanted to see what is affecting Jean-Paul Marat. Now, unfortunately, I don't have a perfect answer for you. So they compared that bacteria and virus DNA to a lot of different organisms. So they found the DNA didn't match syphilis, it didn't match leprosy, it didn't match tuberculosis, it didn't match thrush, it didn't match scabies. So we're like, okay, it doesn't match a lot of that. So we've ruled out a lot of things. Scientists don't have a clear answer for what Jean-Paul Marat was kind of affected with in his last few days, but they can say that it was some sort of fungal infection. The one way they could answer this is if they go back and they take more of that DNA found in that newspaper, but the downside is that they have to be careful. They don't want to damage that newspaper since it's a historical relic and they want to keep that safe for the museums. But that is one way we could find an answer. So I thought this was a really neat example. Usually you hear stories about how scientists are trying to kind of understand disease or maybe develop a vaccine or cure. But this is kind of an example of how scientists can go back into the past and find answers to questions that we still don't know the answer to. So yeah, this was a story that I wrote about. I thought it was super interesting. And I think a lot of people actually found it super interesting because I think over 1,000 people have read this one, which was very surprising to me when it was just a fun story for me to write about. And yeah, that is kind of a picture of what I do every day. I carry out science in the lab, although right now it's at home because we're in a quarantine. And then I kind of love writing stories about science, especially about DNA, to share more of what scientists are doing in the lab with everyone over the world. And with that, I wanted to say thank you everyone for tuning in. If anyone has any questions, feel free to drop it in. And I'm going to throw a question out at you as you all get started, which is, would you consider sequencing your DNA? And yeah, thank you.
Thank you so much, Farah. That was awesome. We have quite a few people joining in. We've got Ryan, Illinois, Mount Orab, Wisconsin on YouTube. That's fantastic. If anyone else is joining and wants to let me know where they're joining from, that would be great. And you guys can type questions in the chat bar. But I want to kick it off because before we went live with this presentation, Farah said, oh, you can ask me this question if you'd like about how to extract DNA from a strawberry. And you can't do a DNA presentation and hint at extracting DNA from a strawberry without bringing it up. So please, I would love if you kick us off by showing how to extract DNA.
I would love to. So I unfortunately don't have a strawberry, but I can still walk everyone through how to do it. So you can actually extract DNA from strawberries, bananas, saliva, although you need a lot of saliva, cheek cells, and so on. Usually a strawberry works best, but you can get creative. Usually soft fruits are good. So step one is kind of gathering all the resources that you need. So number one is a strawberry. Number two is you want to prepare that extraction mixture. And I know extraction mixture sounds fancy, but you probably have everything you need at home. So you need a pinch of salt, just a pinch. You need 2 to 3 drops of dishwasher liquid. So kind of ask your mom, hey, can I have some detergent? Hopefully mom will say yes. And you want a 1/3 cup of water. As soon as you get these 3 things, you want to mix them all up, and that's your extraction mixture. So then you're going to grab a Ziploc bag or any kind of small container. You want to drop in your strawberry. You want to add in 2 to 3 tablespoons of the extraction mixture in the Ziploc bag. You want to seal it carefully to kind of push out all the air. And then you're going to gently crush your strawberry for a minute. So by gentle, I do mean gentle because you don't want to kind of pulverize that entire strawberry. You want to have like break down the chunks, break down the strawberry into parts, but don't worry about kind of exterminating the strawberry. Just enough crushing will be fine. After that, what you're going to do is you're going to pour your strawberry Ziploc bag solution into a glass. You want to avoid the chunks. So if you have like a sieve, like a tea sieve, or just pour slowly, you want to get more of the liquid and less of the chunks. And then this is probably the hardest part, is that you want to find some ethanol. So this can be also labeled as isopropyl alcohol. You can check your supermarket, your drugstore. If your mom or your sister or your sibling kind of has what you call a nail polish remover, that also works. And you want to pour this ethanol, make sure it's cold, so just drop it in the fridge for a few minutes and just pour it down the side of your glass. And then just wait a few minutes. The strawberry DNA mixture is going to react with your ethanol. In a few minutes, you'll kind of see these white, gooey, sticky strands. If you wait longer, you'll kind of see those DNA clumps stick together. So now you're probably wondering, okay, how do we just see DNA? Why don't I see DNA with my eyes all the other days of the week? So the fun thing about strawberries is that they actually have a lot of DNA packed together. They kind of have 8 times the DNA in one single cell compared to us humans. So that's why we can see so much. And I'm going to explain some of the science behind it in case you're curious. So when you add in the detergent to your, what you call a strawberry, so firstly, you're breaking down your strawberry with your hands. You're breaking down those cells. Adding the detergent means you're breaking down the cell membrane. So you're kind of letting loose everything that's inside cells. That includes your nucleus, that includes your DNA, that includes trash, that includes mitochondria, just everything is kind of let loose. The salt means that the DNA is going to kind of come together and clump. So you're kind of narrowing, getting your DNA to stick together. Now, usually your DNA will dissolve in water. That's why we added in ethanol. We want to force that DNA to not dissolve and just become those gooey strands. And that's kind of all there is to DNA extraction. It's something that you can do at home at any time. You can't usually see DNA by eye, but by doing this, you can, see a lot of DNA. And it's really fun just grabbing a toothpick and pulling it all out and be like, hey, strawberry DNA. How fun is that?
Awesome. Very cool. Thanks for the great slides too. So those will all be up on YouTube for anyone who wants to follow along with those steps and hear Farah talk about it. That's fantastic. What I want to do now is if you could come out of screen share actually so we can see you again, it would be lovely to ask questions directly. For people on YouTube, again, don't be shy, ask some questions, people. I know that I would sequence my DNA. And I think a lot of people now are using things like genetic or 23andMe, Ancestry to sequence parts of their DNA. Would you or have you sequenced your DNA?
So I have not sequenced my DNA yet. I'm just not willing to drop $100, but there are a couple of questions that you want to keep in mind before you sequence your DNA. First, you want to make sure you're considering your safety. So how is the company going to store your DNA? Are they going to protect it? Or are they going to perhaps sell the information? Like, these are all important questions that you want to consider. And secondly, I think it's worth waiting before you actually sequence your DNA right now, because scientists are still trying to understand the genome. Like, there's so many parts that we still don't understand. So I would actually say perhaps in another 10 years, if you sequence your DNA, you'd learn a lot more than you would right now. So for me right now, is I haven't sequenced my DNA yet. I'm not going to probably for a while, but maybe one day in the future when I know that kind of all the scientists have learned a bit more and I can get more value for my money, I might give it a shot.
Fantastic. Yeah. Speaking of DNA, so you got a chance to write about this cool mystery being solved of like centuries old DNA. How long does DNA last? Can we get Jurassic Park? Is that possible? Or like, is there a time limit at which DNA starts degrading to where it's not usable for information and inquiry?
So you've kind of touched on a field that's called ancient DNA. There are a lot of scientists who actually study ancient DNA, like the study that I wrote about. So how long, there's no perfect answer to how long DNA lasts. It kind of depends on what it is. For example, let's say I had some DNA left on this Harry Potter book. I am going to use Harry Potter every time I can. So if I perfectly preserve this, like let's say I left it in a cold room where it wasn't humid, maybe the DNA would last longer there. But if it was kind of out in the open in sunlight, that DNA might degrade faster. So kind of the conditions where the DNA is important. So a lot of scientists are working with ancient DNA. For example, the example I gave was scientists literally pulled out DNA from an 18th century a dead person. So it's happening. This is science. It's real life. And about Jurassic Park, so I can't promise that we're going to get T-Rexes walking around right now, but there are actually scientists who are trying to recreate the woolly mammoth. So they kind of found DNA from the woolly mammoth and they're trying to synthetically build it together. So that field is called synthetic biology. So people are either trying to bring back dead organisms, or maybe they're trying to kind of combine DNA from different organisms. For example, I know there are scientists who are working on engineering the perfect strawberry. So they're kind of saying, hey, what if I took a gene from corn and I added to strawberry? Could I make the strawberry brighter? Could I make the strawberry juicier? Could I make the strawberry bigger? Like, these are some of the things that scientists are trying. Now, I do want to point out that for every study that scientists are doing, at least at a university or an academic institution, they have to get permission first. We're not just going around and bringing back T-Rex. There are a lot of steps involved and you have to get permission before you do a study, and not all studies work. So I'm excited to see the woolly mammoth, but I don't think we can expect results for a while, sadly. So you're gonna have to keep that little dream of Jurassic Park just a little longer.
I'll just keep reading the books. You have your Harry Potter, I have my Jurassic Park. Together, some great pop culture. You mentioned earlier in your talk, and I want to harp on it, you mentioned that some people share both epilepsy and autism spectrum disorder. Is there a percentage of people? Is this something that, like, if you have one, you're more likely to have the other than the general population, or how does that work?
So we do have numbers on how often this can happen. So for example, people with epilepsy, like around 6 people with epilepsy tend to have, 6% of people with epilepsy tend to have autism spectrum disorder. And in autism spectrum disorder, it's like, I think 5 to 44% of people with autism spectrum disorder may have epilepsy. Thing is, we don't have solid numbers. It's because usually when physicians or researchers kind of work with people with epilepsy or autism spectrum disorder, they're always focusing on one group. So they only work with people with epilepsy, or they only work with people with autism spectrum disorder. So we kind of need more researchers, more physicians to kind of work with both groups so that we can better understand it. So we're still learning a lot more about how often you can see them or whether there's increased risk. Like there are a lot of questions, which is why I'm still researching and trying to better understand the two disorders.
And with this sort of work, so I mean, I've certainly heard about it with regard to some more physical disorders, things like cystic fibrosis, where genetic therapies could eventually lead to getting rid of that as a disorder that people have to deal with. I mean, we're in the very early stages of research now, but I mean, is there a theoretical possibility that once we understand more about things like epilepsy, we could make sure that people don't have to suffer with that, or is that, I don't know, that the goal of the research or not your research, or
It's not a goal of my research. I think it would be very hard for epilepsy and autism spectrum disorder. So there are a lot of different kind of changes in our DNA which could lead to epilepsy and autism spectrum disorder. So it's pretty difficult to even consider something like that. For some of the more severe types of epilepsy, there are anti-epileptic drugs which can help people. So it really depends on kind of the person in charge. And obviously, not a medical doctor, I can't give advice,
Right.
but then there are disorders like cystic fibrosis where we know exactly what, so we know that changes specifically in this gene called CTFR will lead to cystic fibrosis. And there are scientists who are working on therapeutics, and I think some of them are getting further ahead. So it really depends on kind of the disorder. If it's kind of one change will lead to the disorder, then yes, there are a ton of scientists who are working on therapeutics. But for things like epilepsy and autism spectrum disorder, there's so many changes that could lead to it, and we're still like trying to get our heads around understanding it, that there isn't going to be one cure that kind of explains everything. And yeah.
Well, this is perfect, actually, because I mean, I think a lot of people, when they're watching the news and hearing about DNA work in just public understanding of science pieces, this idea of a gene for something gets brought up a lot, or there's one gene, or there's one solution. And in the vast majority of cases, it is more complicated than that. Is that a good statement generally?
Yeah, that's perfectly true. So there, like, as I said, there's so many letters in our DNA. We're just beginning to understand what it all means. And like one change could lead to a disorder, or it could lead to nothing, or it could lead to or it could be multiple changes that lead to a multiple number of things that show up. So we are like, we sequenced our entire DNA in 2003. We're now in 2020. But we're still trying to understand the DNA because there's so much to learn. So you're perfectly right. Yes, we are still understanding it, and it's not one change in one gene that can explain answers.
Yeah.
And yeah, DNA is huge.
Inner world is complicated. It's exciting to be in that area of research, because I mean, there's so much to learn, and we're just scratching the surface of it. As you said, 2003, like as far as major scientific discoveries go, that is incredibly recent. I mean, that is within most of the lifetimes of people who are watching this, and maybe recent history for some of our younger kids today. But that's thrilling. You talked about science policy, and so I mean, this is a broad thing and a question to ask, but are policies for science improving, whether that's in Canada or worldwide? Are we understanding the value of science? Are we starting to support it more? Is this a crapshoot? I don't know, anything you can share about your feelings on that?
Sure. So I think right now with COVID-19 and the pandemic, we're seeing science happen in real time. So we're seeing countries all over the world giving more money to scientists to build vaccines, giving more money to public health so we can do things like quarantine and take care of people. So I'm seeing increased support for science. The sad thing is like, if we had kept this support for science throughout the years, then maybe the pandemic wouldn't have been as bad. Maybe scientists would have had more money. It would have been easier to jump into action to build vaccines. So there's good news and bad news. I'd say it's great to see science in the news right now. It sucks that it had to come to a pandemic for us to see more science in the news, but there is support. And I hope that when this pandemic does end, we can kind of continue to see the support. So if you're kind of tuning in right now, my kind of question to you is like, what parts of science are you interested in? Because all the parts of science kind of coming together for this pandemic. For example, there are genetic scientists like me who are kind of off sequencing the virus to understand, hey, why does the virus affect people differently? Then you've got public health folks who are saying, this is the next step we should take. We should end quarantine. We should start quarantine here. So like there are so many different parts of science that you can get involved in, and that can help one day in the future, hopefully not in a pandemic, but in simpler, calmer times. So back to your question, yeah, a lot of countries are doing a lot to support science. It's very exciting and inspiring to see. I just hope that we can continue to see the support when the pandemic ends.
Nice. One of the questions that we get a lot here is this idea of how much does science cost, or how much does it cost to put rockets in space? And I always like to highlight in these questions that, it does cost a lot. It costs a lot to fund research agencies of major countries. It costs tens, if not hundreds of billions of dollars. But comparatively to the countries, the amount of money that they produce as a nation, it's a really small percentage of what countries produce that goes towards that sort of research and development. And it leads to things like enhanced communications, understanding our genome, exploring the solar system, fighting pandemics and more. So much good comes out of investment into science that it is always a pursuit worth, going headlong into it as big as you can. So I'm so glad we got that question. All right, Zeke and Joe in Mount Orab, Wisconsin, they want to know, can you change your DNA? So I'm taking this as like an individual. Right now, if I wanted to change my DNA, could I? Is there a way to do that?
You can do it temporarily.
Okay.
But so you've got DNA all over the cells in your body. So if you want to change your DNA, you technically want to change it in every single cell of your body. Right. That's a lot. That's like millions of cells. There are so there's something called CRISPR, which is a gene editing system. So CRISPR is like a pair of molecular scissors. You can kind of go and snip out DNA where you want, and remove it, edit it, change it. So there are some scientists who are using CRISPR to help in cancer treatments. So as immunotherapy, where they kind of target cancer cells and they change the DNA inside cancer cells and help fight cancer. But that's very specific, like the scientists are targeting cancer cells to make it happen. So that's one way you can change DNA. Right now, no, there aren't perfect full ways that you could change your DNA. Maybe that's something in the future that we can see. So things like Spider-Man getting bitten by a radioactive spider and that changed his DNA makeup so that he could become like a spider. That's, I think, way off in the future. But there are exciting people who are kind of working on things like CRISPR and how to edit your genes. So it might happen. But again, it would need a lot of testing in animals and lab rats in clinical trials before it ever becomes common in the real world. But if that's something you're excited in, then I think you should check out, like you should first definitely go into science and university, learn more about our DNA, check out CRISPR, check out all these fun things happening in synthetic biology or ancient DNA. And maybe one day I'll be able to say, hey, Zeke and second person whose name I can't remember, they actually found a way to edit our DNA. Wow, cool. So maybe you'll be the ones who do it.
I'm so glad you brought up CRISPR, and this is something that we're just starting to touch upon in our sessions here at Exploring by the Seat of Your Pants. But this idea that we have the most elaborate and complex tool for excising and changing DNA ever. And this is something that we can do with individual cells really readily, but our bodies are made up of so many that is now, for now, too complicated to do in a serious fashion. But again, look at how much we've gained in the last 17 years since we sequenced the genome. And so the next 20 years are going to be the new frontier of biology. And with that serious comment made, I also want a screen share of you going like Spider-Man, because that was amazing and no one's ever done it before. All right, we're amazingly nearing the end of this. So I want to wrap up with two questions that I always love to ask. What is your favorite part of your job? You have so many roles. Is there anything that really like you do it and every time you do it, just you're over the moon?
I think I love talking to people like this. I think that's always my fun part because I just get to talk about science and no one can stop me and it's lovely. But I think especially I love doing strawberry DNA extraction because it kind of blows people's minds how I can take a strawberry and just pull out DNA. It's just like, what? Strawberry has this? we can do this all the time. So I think talking to people about science and maybe just strawberry DNA extraction is kind of my magic trick at parties is what I love to do.
Awesome. Well, we're certainly happy to have you. And it's funny how often the answer to that question is talking to people. I mean, I think a lot of people think of scientists as people that, sort of want to be doing stuff in the background. And increasingly, our generation of scientists really love to share the work they do widely and really inspire people and take selfies. So, combined, we're all set. And I really appreciate you being here today. Okay. If people, so you've highlighted all these amazing facts. And this is only a 40-minute session today. So if people want to finish this and go and learn more about DNA, get excited about it, where can they go? What are some great resources that you'd recommend? Books, websites, other scientists to follow, anything that jumps out?
Nothing is coming to the top of my head, which is bad, but I would recommend checking out other scientists. So there are a lot of incredible scientists who are doing work here. So there's, of course, Samantha Yamin, Science Sam on Instagram, who's always sharing fun science facts with the world. There are groups like Genome BC, so they're a group here in British Columbia. If you actually check out their website, they've got a bunch of DNA activities. So you could try the strawberry DNA extraction, or you could try more complex things. For example, I know they try to build a DNA model using pipe cleaners. So if you want to build your own DNA model, that's one place to go. And there are a lot of incredible books on science out there. One of my favorites is kind of 100 Women in Science. So it's by Rachel Ignatowski. I know I've gotten her name wrong.
Yeah, I love this book too. This is an amazing book.
She's kind of like drawn 100 scientists, and she's got kind of these amazing descriptions of them. So if you want to learn more about scientists, that's definitely one. And I guess my last plug would be to go learn more about Rosalind Franklin. She was actually one of the scientists who helped discover the structure of DNA. A lot of the credit goes to Watson and Crick, but Rosalind was right there doing the work. So she's kind of the DNA lady. She pioneered everything that I'm talking about today. So definitely go learn more about Rosalind Franklin. There are a bunch of documentaries and books about her. She's incredible. She deserved that Nobel Prize. Still mad for her.
We're mad for too. You're the first person ever to bring up Rosalind in any of our programs. And like Rachel Ignatowski's book, where it's just beautiful imagery, beautiful science illustration to highlight this amazing work. I know there's a Rosalind Franklin book about this, and I cannot for the life of me think of the name of it right now, but do check it out. She came up with the imagery that enabled Watson and Crick to make that big discovery, and they basically stole the stuff that she had. So she's awesome. Fantastic. This is great. And I'm trying to think of other books myself. I'll put them in the chat bar if I think of any. But before we wrap up, and I know you had a slide about this too, how can people find out more about you? You're doing all this fantastic stuff. People finish this session, they want to learn more and keep track. Where can they go?
So I am going to drop my slides open and go back. So yeah, you can find me on Twitter. So at This Is Far, or you can check out my website, farahqaiser.com. There is a fun DNA zine that I've got there. If you want to learn more about DNA, if you want to do the DNA extraction, check out the resources section, and you can kind of do that in your own home, or maybe even download it and just read it off to your parents, and they'll be like, where are you getting these facts from? And just be like, I learned it from exploring by the seat of your pants, Mom.
Fantastic. Also, the first ever use of the word zine in any of our programs. This is just the most unique session of all time. I love it. All right, this has been great. Thank you so much for joining us today. I certainly learned a lot, and I'm sure everyone joining at home did as well. Do check out those great resources. Get some books. Look online. Look up CRISPR. So this is CRISPR. Just ignore the E. And you'll learn so much about some of the most cutting edge research in the world told by some of the best science communicators online. Farah, with it, like what we, I just want to say thank you so much for joining us, and
thanks for having me.
Hope to have you back soon. This was great.
Yeah, bye everyone. Stay safe, stay home.
Stay safe, stay at home, and we'll do another one of these when all the kids are back in school. We can have 250 people all asking you a whole bunch of questions. Without further ado, guys, have a lovely rest of your day.